Okay. Hello, everyone. Welcome to Right Science Club. The Rights Club is an initiative that aims to raise awareness and provide training in reproducible, interpretable, open, and transparent science. You can see in our website and that we have a fantastic set of speakers and events lined up. And please check out our next speakers and the summer schedule on the slides. Our events are entirely free and open to anyone. If you cannot make a particular event or would like to keep track of upcoming events, please check out our website, Twitter, OSF, and the YouTube channel. You can also subscribe to our mailing list. Finally, we're currently recruiting new members who want to join us, and if you're interested in spreading open research, please send us an email. And before we start, just a bit of housekeeping before we introduce our speakers. We host our events on Microsoft Teams live events, which has a fantastic auto subtitling feature, and you can ask your questions via the Q&A message box. This will be moderated by Lorenza and I during and after the talk. Okay, now let's move to the talk. Our speaker for today is Guiomar and Rotem. Guiomar is the senior researcher at Indiana University Bloomington. She's also the chair of the BIDS steering group and a collaborating researcher in cyber BBN spin. Rotem is a postdoc researcher at Dartmouth College. They have been working on promoting topics related to open science, and recently they just published a preprint about how to do open and reproducible neuroimaging studies. Me, as a junior researcher who is also working in neuroimaging and child psychiatry, even I'm quite familiar with open science, it is still sometimes hard to find the best instruments to improve the transparency and the reproducibility of my work. Today, Guiomar and Rotem will give us an overview of the community-developed resources that can support collaborative, open, and reproducible neuroimaging in the entire research cycle, from inception to publication, and across different neuroimaging modalities. Okay, that's it from me. Guiomar and Rotem, the floor is yours.
Thank you very much. I'll share my screen. Okay, can you see it well? Guiomar will start, so I'll let her take the stage.
Thank you very much. It's my pleasure to be here today together with my colleague Rotem at this Riot Science Club. And I really want to thank Lorenza, Bing, and the team for the invitation and organization of the event. In this talk, we will present our work on open and reproducible neuroimaging, going from the study inception to publication and beyond. If you are attending this seminar, you will probably be familiar with open science and practice and aware of its importance in science. However, as you may know, the adoption of open practices remains limited in researchers' day-to-day work. A recently study by Parrett and colleagues showed that only 42% of respondents have never pre-registered a neuroimaging study, and 34% have never shared their raw neuroimaging data. Open science practices have many benefits for individual researchers and scientific progress. So, like mitigating transparency and reproducibility issues, and also at the individual level, increasing exposure, reputation, chances of publication, and potential collaborations and funding opportunities. So, indeed, new policies by funding agencies are starting to emerge to support open practices and tools. And yeah, as we were saying, indeed, there's an abundance of tools, which is really promising, but also a bit challenging, since the information can be scattered over many sources. And this makes it difficult even for experienced researchers to navigate this complex ecosystem of available tools to make sustainable choices. That's why a group of over 20 researchers from different labs around the world already, and which we are actively developing or using open science tools in neuroimaging, joined together trying to provide an integrated overview based on our experience of the different resources available at each step of the research cycle. We release a preprint at OSF that you can freely read it if interested. And here in this talk, we will try to summarize some of the main points. Yeah, as we were saying, in this work, we aim to provide an integrated overview of community-developed resources that can support collaborative, open, and reproducible neuroimaging throughout the entire research cycle, going from inception to publication, and across the different neuroimaging modalities like MRI, MEG, EEG, PET, and so on. So the talk will be, the content is very heavy, so you are completely free to go to the preprint to go for dive in on further details.
Thanks, Yara. Okay, I just want to say one thing, that this is the first time we're doing a joint talk, so bear with us, you know, if the transitions are not as smooth as they should be, but we'll try to do our best. And I'll start with the first part. So obviously every study starts, you know, with the idea and the preparation, and it's important to take into consideration exactly how you want to do it and what, you know, how reproducible you want it to be, because many choices that you're going to make at the initial steps would affect whether or not you could, for example, share your code later or can actually, you know, reproduce your results. So the first part is to prepare for the study. And usually, not always, but usually people do a pilot study before, where we collect data that are not going to be used to actually, you know, test the result at the end, but are just there to see that we could collect the data, everything is working properly, participants understand what they need to do, the experimenter understands what they need to do, the analysis, you know, all data are lost, we don't lose anything in the way. We could synchronize, for example, the stimuli with the brain data, you know, the behavioral of the brain data. The analysis that we planned can actually be done. Many times pilot data are used for making sure that the behavioral effects, or at least the trends, are there, you know, descriptively. You can see the effect if you're looking for the brain mechanisms. And sometimes people use open data or simulations to pilot their analysis, but obviously there's, you know, nothing like the actual data collection process to make you realize what you did wrong, because there are always errors and things you couldn't predict. So piloting is very important. It could also be used for power analysis to, you know, decide on sample size. The next step is to, you know, plan what you're going to do and document everything. So, you know, once your code is ready and everything is ready to run, it's best practice to have kind of a standard operating procedures, an SOP, where you just document how the experiment is going to be. You know, what you're going to say to the experiments, how you connect everything, what you do when, where the data are stored at the end. Even recruitment could be part of this, which is really useful. And obviously these things change during the project. You know, if suddenly you understand that participants are not understanding something, then you could change, you know, what you're telling them. If you understand that things are taking too long, you can change something with the timing. If you see that, you know, some machine fails and you need to make sure some connection is correct, you can add a picture of how it needs to be corrected. So things like that are very beneficial. And ideally, you know, the best scenario is if you could take, you know, a new experimenter or someone else from another lab could just take this SOP and, you know, replicate your experiment perfectly with minimal guidance. This would be the best, and this requires a lot of details, because there's a lot of things that the person who plans the experiment and code everything knows that, you know, another person could not know. And so this is very important and helps to have better quality data and making sure you could actually, you know, test your research question with the data you get. Ideally, this could be shared with the dataset of the publication. It doesn't happen frequently, I would admit, but it would be good if it would happen more. And you can find examples of SOPs or other protocols in these places that are shown here: the Protocol Exchange, Protocols IO, and Nature protocols. The next step, which is important to do prior to you know, collection of the data, is the preregistration. And preregistration is the specification of a research design, hypothesis, and analysis plan prior to observing the outcomes of a study. Ideally, it's before data collection, but sometimes it can be because the data are already there, or you're using shared data, which is okay too, as long as you don't know the outcome already. Because the main purpose of preregistration is to distinguish between exploratory research and confirmatory research. And in confirmatory research, you need to have an hypothesis before you're looking at the data, because you're using the data to test the hypothesis. While in exploratory research, you can generate new hypotheses and just, do whatever you want with the data, just to look for effects. But obviously, you cannot, generate and confirm an hypothesis on the same data. And I want to put it up front. Both are extremely important for science. Both are needed. We need both exploratory and confirmatory research. It's just important to know which is which, because the statistics are a bit different and the conclusions you could draw for each of them is different. Okay, so if the data were seen and then the hypothesis was generated and tested, then it's not a real test of hypothesis. To test an hypothesis, you need an hypothesis before you do the test. So this is important for registration. And pre-registrations are usually posted on a public registry. There's timestamps that are usually frozen read-only, so changes couldn't be made. Of course, sometimes you do want to change something in your pre-registration. So what you need to do is just to post another version of the pre-registration so people could see both, and you can explain what was changed and why and when, and everything is timestamped. The three main platforms for pre-registration are OSF as predicted and Clinicaltrials.gov. OSF is probably the mostly used one. It's large. It has many different templates, a lot of information, many types of different pre-registrations. And you can really use it to learn more about pre-registrations as well. And you can connect it to your data. So you can put your data that you're sharing or other files, and this is really useful. As predicted is much simpler, just straightforward, less questions, just the minimal information you need to make sure someone predicted what they're testing. So it's much easier to use it, and there's also some useful information there to be used. And Clinicaltrials.gov is a site operated by the NIH, the American National Institute of Health. It's mostly for clinical trials, as you could probably figure out for the name, but actually NIH now asks funded projects that are not exactly clinical trials, but have some kind of a manipulation related to health to post things there. So it's also being more and more used. And pre-registration is great. It's, it improves transparency. You're very transparent and clear about what you planned before and what not. You will not say in your paper as predicted if it wasn't predicted, for example. It will prevent most of the p-hacking and erking, consciously or unconsciously. I assume most people here know what it is, but I'll say it briefly. So p-hacking is when you try a lot of different things and just report the one that yielded a significant result. And erking is when you change your hypothesis after the results are known, which you could see here in the nice illustrations. So we want to prevent it because it just reduces the replicability and the robustness of results. It also improves planning and helps spot mistakes in time because you can coordinate with your collaborators before you post the registration. So you could notice if something you can't really test what you wanted, or you're missing a covariate that you're not collecting or something like that. And also you can get some recognition. Now many journals would give you a badge for pre-registration if you pre-registered your study, which is kind of nice. Taking it one step forward, the registered reports format is kind of an official pre-registration. There are hundreds of journals that allow you to do such a thing. You could submit your research plan, so it's kind of an introduction and methods and prediction before you collect the data. So before you know anything about the results, you just submit for peer review your plan. Then it goes for stage 1 peer review, and if it is accepted, you will get an in-principle acceptance, which means that as long as you're going to do what you said you're going to do, then it's going to be published in this journal. So once you get this in-principle acceptance, you can go collect your data, analyze them. You could do analysis that was not planned, as long as it's flagged as exploratory analysis. That's great, no problem, but you need to do what you said you're going to do too, as well. And then after you have everything ready, you write the paper and submit it for stage 2 peer review, where they will focus not on whether your results are significant or cool or not, but whether you did what you said you're going to do and whether your conclusions make sense based on what you got. So this is really improving science because it helps you, as long as your research question is interesting, then any result should be interesting, all right? As long as your methods are valid and what you're asking is important, and the journal give you this stamp at the first stage, then it doesn't matter if the results are significant or not. It should be interesting anyway. And in other venues, it's hard to publish such results, but here you will be, you could publish it even if it's a null result because it's interesting and you did a good study. So I encourage you to use this format. Another thing you should plan is the ethics and the sharing plan. So you need to consider all the privacy issues. For example, if you have videos of people that you will want to share later, there's a lot to think about there because it's identifying information. Specifically in neuroimaging, it's important that in the informed consent of the participants, they will consent to share their data, even if it's identified, because otherwise you will not be able to share it at the end, and then you will regret that you didn't do it before. So it's important to think about it beforehand, and the Open Brain Consent initiative is a really great one. It has a lot of information about legal stuff and what needs to be included and how you could phrase it for participants to understand what they're consenting to. And they have many templates in many different languages. So definitely check it out if you're going to start data collection. Make sure participants are consenting for you to share the data as you are planning to do. And the last thing in the preparation is to monitor the quality. So it's important to not just collect your data. You could collect 100 participants and just then start to analyze things and realize that something was completely wrong, and then it might be too late to fix it. And there are going to things are going to happen unexpectedly. There are errors all the time. Experiments are getting more and more complex with more and more machines and people involved. So things will break at some point, and it's important to make sure that you detect it in time, so you could actually improve things and change them. If something is not logged as it should, if something is not synchronized, if something does not happen when it should, if participants fall asleep all the time, if they move too much, if something is not working well, you should detect it early. So quality control checkpoints are a great way to do this if they're planned in advance. For example, you could look for artifacts. For example, there are some artifacts that you will not be able to see after you did like skull stripping in an MRI. So you want to look at it before the data are already processed. It's important. I don't know, if it's EEG, you want the participants to blink and make sure you see it, because otherwise something is not okay, right? If participants are pressing buttons, you want to see that you get a motor response. In MRI, you can't see it online, but at least look at the data after a few participants and see that. So this is important. This could happen manually, but there are also some softwares that could help you automate this. For example, MRI QC. And all the examples here are MRI, but I think things are starting to be developed for EEG and MEG2 and PET probably. MRI QC allows you to do quality control for MRI data. Quality helps to do it with surface data. Yeah, so it's really useful, and there's many people working on trying to automate these processes by telling you which one, for example, which participant need to be excluded, but it's really hard, so it's not yet automated. So you do need to decide in advance what are your thresholds for excluding participants, for example, which should be part of your pre-registration. It's also good to have a positive control analysis, which is not the effect you're looking for, but effects that should be there if everything is okay. So I gave one example before. If participants are pressing buttons, you want to see a motor response where they're pressing the buttons, right? Or if they're seeing something, you want to see a visual response. So this is very useful to test all your pipeline on a positive control analysis that you need to see an effect before you're actually doing something for your own data, and then you can improve the pipeline to make sure it's good before you run it on your specific outcome that you're looking for. Because if you're running it in your outcome and then changing things, this could be p-hacking, because you could just change things until you get an effect, even if you don't really need to, but we're human. The next part and the research is data acquisition. So once you're ready, you have everything planned, you can start collecting data. This is very technical, so I won't go too much into details. I'll just mention a few tools that could help. So the main issue in neuroimaging across modalities is that they're all based on vendor systems. So the software and hardware are closed. It's not open source. You can't see what's going on exactly. And many times participants, we get what we call raw data. For example, in MRI. Sorry that all my examples are MRI based. I'm from MRI, but Guiomar is from MEG, so maybe it will balance. Yeah, what we get and called raw data, like the DICOM files, are not really raw. A lot of things are happening in the scanner before we get these reconstructed images. So this is important because it could be different between scanners and between different companies, vendors. And such heterogeneity could affect the result. It could lead to spurious, non-biological differences. It's even more important if you're using data from different sites, but it could be also important within the same study on the same scanner. So many great people are working on that. They're developing open tools to overcome some of these problems. One huge problem is that the sequences could be different. Acquisition sequences would look the same, but might be different across scanners. So large projects like HCP, the Human Connectome project, for example, they were able to have like consensus protocols that could have been used on different scanners. It was all Siemens specifically, so it was easier, but still different types of scanners. But other people are working on like an already developed vendor-neutral acquisition sequences. You could use, for example, PulseSec or PyPulseSec to do something like that. There are also tools for reconstruction frameworks, open and vendor-neutral, and tools for quantitative MRI, and that could be better, compared between sites. And many other things are also happening in EEG and MEG now. So these are cool developments that maybe allow us to do actual open science from the beginning to the end, and not just after we get the raw data. And because of all of that, it's really important to report in details exactly all the parameters you used and the exact machine you used, because it could make a difference. It's also important in behavioral part. So when we run a task and collect data and show stimuli, it's important to do it in a way that could be reproducible. There are great open source software for this. For example, there's PsychoPy in Python or in MATLAB Psych Toolbox. There are some modality specific. Repositories could help you with videos. Precore could help you for EEG. So this is very useful. And again, it's best practice to share these things so other people could replicate your experiment and also, understand exactly what you did. And if not, at least very thoroughly describe exactly what you did. Yeah, and I will switch to Guiomar again.
So in this section, we will show open resources for research data management, and this refers to how data are organized, maintained, annotated, tracked, and stored and accessed throughout the research project. And in this section, we have divided in 3 parts. The first is the standard and organization of the data, then metadata and annotation, and finally the data management and tracking. For data organization and standards, what happens? Data is organized differently within institutions and even within a lab. So this produces a lot of misunderstandings, waste of resources, and poor reproducibility of results. So the use of a standard is really, really useful. The main standard in Neuro-Imaging is the brain imaging data structure, which is a standard to store, organize, and share Neuro-Imaging data. And it's based on very simple file formats and folder structures. And it has your data files together with your metadata. And the metadata should be readable by humans and machines, like in this example. Rotem, if you can click twice. Yeah. So you have your folder organization and then your data and your metadata, and then the metadata is like organized in fields and subfields that you can are standardized to describe your data. And there are also multiple tools to make it easy for researchers to incorporate BIDS into their current workflows and to maximize data sharing opportunities. For example, BIDS converters that are able to convert your raw data into BIDS standard, and also a BIDS validator, which ensures your dataset is compliant with the BIDS specifications. And also there are some general tools like for interacting with the data, plumbing, querying, like PyBids and BIDS MATLAB. And finally, we have a really good set of BIDS apps, they are just analysis pipelines in a container, which accept BIDS dataset as an input, and it's really convenient to standardize your analysis workflow. The BIDS was created originally at Raspoldrack's lab at Stanford, and it was led by Chris Gorgolewski, which initiated this effort for MRI. And soon we extended it to MEG, and other modalities came like EEG, intracranial EEG, genetics, PET, microscopy, which have been really recently released. And there are many more extensions on their active development. And I want to really say that BIDS is a very open and inclusive community, and any of you that are interested are very welcome to join and contribute and to extend the standard or to or to provide some feedback. And regarding metadata and annotation without a standard, it's also very cumbersome and error prone. So it's really important to have some standardization for metadata and data annotation. For example, the hierarchical event descriptor, HID, it's a structured vocabulary for annotating data, which is human and machine readable. And I have to say that this is also fully integrated with BIDS and can be extended by other supplier schemas. And as an example, here we can see how we will annotate the presentation of a face image. For example, it's a sensory event. It is a visual presentation of an image that has face and hair and so on. So there are tools that are able to span these paths and this control vocabulary into some hierarchical relationships. And if we see in the next slide, we will see the full path of the sensory event, visual presentation, image with face and hair, but you see the full path that allows you to have some hierarchical structure that allows you further analysis and categorization of your results. In the same way, there's another initiative. Well, this is a really broad initiative, which is the Neuro-Imaging data model that many throughout the research process, you will see that they have many tools available to deal with reproducibility issues, and they are a very nice open initiative, and it extends the provenance standard for Neuro-Imaging. And they use this provenance information to link components between the different stages of the process. So in particular here we wanted to mention the NIDM terms, which is an effort that collects community-developed control vocabularies and techniques for associating concepts. For example, Interlex or Cognitive Atlas or BIDS vocabulary. So it has like search database that you can enter a term, see how it's defined and which relationships has with others. So it's really useful tool to search across publicly available datasets. And regarding management and tracking, so many files change over the course of our projects. So we need a way to smartly track them. So there are some initiatives like DataLab or BrainLife that allows you to smartly track these changes. In DataLab, which is a really useful initiative to track your data, it allows to manage and version control digital files in a decentralized way. It tracks the data and metadata of any file on any type or any size in a scalable repository structure. It can be on your local computer, on the cloud, or on a server, and it can retrieve it very smartly. So they have a data lab handbook, which is really useful to start using it. And they also have here a little example, which I won't go through all the details here, but I have put the link, and you are very welcome to just join. Just read it carefully and see how the different steps of setting up a data lab structure and how to use the tool and this resource with your local files. Another tool for data management and tracking is BrainLife. BrainLife is an open, non-commercial cloud platform for reproducible data management and analysis, and it tracks data provenance automatically. BrainLife is more based on GIS, on user interfaces, and it allows you to manage your data, your processing apps, and your derivatives. And it has some visualization tools and also the ability to publicate your results linked to each of these data and processing apps. Here you can see a schema of, in the next slide, you can see a schema of more or less the structure. But as we were saying, you have your data sets, your apps, and it also has some free resources to run on their servers, or you can also put your servers in the project and use your local servers if you don't want to use the shared resources. But it's really a nice tool to keep track of all your data files and their derivatives and pipelines. So
yeah, we're going to switch again. Thanks, Guiomar. So the next stage would be the pre-processing and analysis of your data. And there are so many tools, really open tools available. For example, in MRI, the most popular tools are open tools. For example, AFNI, FSL, and SPM, which are more general for pre-processing analysis. Sorry. And there are also some specific tools like ANTS for registration, or FreeSurfer for brain surface. And there are many similar tools in MEG and EEG and PET as well. So this is amazing. There's like so many tools to choose from, and you can make your choice about which one works best for you or for your type of data or for your lab. But given there are so many different tools, many times we need to do some kind of a workflow engine, like just connecting the different steps of the analysis. So this could happen with just scripting. You could do best scripting or scripting, MATLAB, Python, whatever you're using. But there are also some neuroimaging specific solutions that were developed, like NiPipe, which is very useful and very reproducible and open. And there's a huge community around it. So this could be a really good solution to do this. And also lately, there are many attempts to standardize workflows. It started with MRI, but it's definitely going into other modalities now. So for example, in MRI, you have CPEC, which is for resting state, where you can choose exactly what you want in the pipeline and just run it from the beginning to the end. There's MRI QC that I already mentioned about quality control. fMRI prep is very widely used for pre-processing of MRI data. And it led to many similar projects like NI Neuro-Imaging prep, like for diffusion data, for structural data, for quantitative MRI, for many other things. And in MEG, there's the MEG-BIDS pipeline, which is also very standardized workflow. And also, projects like the Human Connectome project have developed their own tailored official workflows and software, which is open and can be used for their data set, which is also very good. All workflows are transparent. They're not black boxes, so you can read and know exactly what's happening. They yield reports that show you what happened and how things look like. You should not trust them blindly. You should check what happened. Look at your images, make sure thing worked properly. They are completely reproducible because as long as you say, the version you used and the data you used, you could just click, just run a single line of code usually to run it and get hopefully the same results. They're automated, which is also very important now that the sample sizes are getting larger and larger. It's really hard to do all these things not in a non-automated way. And many of them rely on containers, which is kind of a virtual machine that allows you to, that you won't need to install all the different software that are used and the exact version and make sure they're all working and the packages are correct. This could be very hard. So the container just takes everything for you, take the environment you need, put it on your computer or your cluster, wherever you're doing it, and just runs it, which is, once you get a container installed, it is much easier and very, much more reproducible. So this is really great. Yeah, okay. But even though we have all these really great automated tools, many times you will probably find yourself needing to write your own code to do something or implement something. And one issue in the field is that most of neuroimagers were not trained as software developers or programmers, and this makes our code less good as people who are professional in code writing. We need to be good in so many things. It's really hard, right? But because code is so important and it can yield to very, very major errors, it's very important to make sure you're doing it as good as you can. It also could lead to biases because people are more likely to check their code if it didn't yield the result they were looking for. But if you get the effect you were looking for, you will just be less likely to check your code again. But there could still be very major errors there. So it's important to make sure that you're doing what you can to make sure the code works properly. One very good practice is version control. There are many available tools, most of them based on Git. So there's GitHub, which is a user interface, and GitLab, DataLab that Guiomar talked about. This helps you to just have versions of what you did in your code and see what was changed and have a comment about why you changed it and when. And it just helps the code being much more organized. And it also makes clear which version was used, and you can release it even with a DOI, with your paper, so that someone could just take this code and use it exactly as you did and reproduce your results. Documentation is key. So I don't cite the source because I don't remember where I heard it, so I'm sorry, but this was such good phrase. So, you know, your primary collaborator is yourself 6 months from now, and your past self does not answer emails. So, you know, if you take code you wrote 6 months, 1 year ago and try to understand what you did, it's often not that easy. So, you know, understanding someone else's code is like a complete mess usually, but also yourself. So I strongly encourage you to document, to give, you know, variables proper names that you can understand what's happening. And it's good to test on yourself, even after a few days. If you could read it and understand what's going on, this is way better than, you know, if not. One thing that is completely lacking in the field is testing. Again, because we weren't trained on this, but there are great tools to help you test your code. It's a best practice to, you know, make sure that, you know, you input something, for example, with or without an effect, and you get something that makes sense. You know, simulate some things to make sure that what you get makes sense, so you could trust what you're doing. It's also really good to let someone else look at the code, but obviously this needs, you know, help from someone and some resources. So if possible, this is very good. There's like code review process where other people are going over your code. This always happens in, you know, IT companies and companies that sell something because they need to make sure that the product is working. We should treat our code, you know, as seriously probably if we want to make sure that our science is correct. And also sharing your code, even if it's not perfect, even if it's not, you know, you're not a superstar software developers, this is very, very much encouraged, and Guiomar would mention it later too. For the statistical modeling part, this is kind of lacking in the current pipeline, standardized pipelines. So there were a lot of effort on like pre-processing and some kind of analysis, but for example, in MRI, the general linear model and how you specify it has a lot of variance between different tools. And this is a stage where many people are writing their own code or doing things manually. So a recent project that's trying to overcome this gap is the BIDS stats model. It's a model specification based on BIDS that just also tells you how you could specify your model in a BIDS way. So, you know, to know which variables you want to put in, what level of analysis participant run, the entire group, which contrast you want to use, which tests you're going to perform, what covariance you want to put in the model. All of these, there are specifications that are going to be released. I think maybe in OHBN they're going to be released. So they're just going to tell you how to do it in a JSON file so machines can read it. And then other tools like Fitlins, for example, which is another open tool, could read this JSON file and actually run the model, feed it to the data. And there are some platforms like Neuroscout that leverage the Fitlins and BIDS stats model to help you reanalyze some open data sets of naturalistic stimuli. So this is based on that. The only reason they could do this, you know, platform and help you do it on the computer is because they could specify the models in a way that would be read by Fitlins and the model could be fit. So this is really, really useful and new, and hopefully will gain popularity in the field. So we talked a lot about the abundance of tools, right? There are so many tools, so many things we could do. This is awesome, but it's also challenging because there are so many analytical choices that you need to make during the analysis. You know, which steps to do, what parameters to put, which software to use, what environment, what package, what version. And these decisions might be arbitrary in a way, but they could potentially affect your results. And this matters because, you know, if a result is very dependent on specific choice that was made, then maybe it's not robust, and maybe it's not as generalizable as we would have wished. We actually tested it 2 years ago. We published a paper where we took the same fMRI data set, we gave it to 70 independent analysis teams around the world, and asked them to take this data and test the same 9 predefined hypotheses and report back to us, you know, the results, statistical maps. And I won't go into details about this project, but what is important here is that we found that the analytical variability was extreme. So 70 teams, they chose 70 different analysis pipelines. There were no 2 teams that chose the exact same pipeline. Again, same data, same hypothesis. So this exemplifies, you know, how much variability there is. And importantly, it affected the results. So different teams reported different results depending on which analysis pipeline they used. So this is very important. And it actually shows a problem that will not be solved by all the great tools that me and Guiomar just showed you, or are going to show you, because even the, you know, a study that is well planned, fully reproducible, everything is safe, the code is perfect, the analysis, everything is just perfect, and the code is shared and everything. Still, you don't know if the results are, you got this result just because of specific analytical choices you made, or a few. Maybe if you would use a different software, something else would happen, right? So even if it's pre-registered, you did everything, it's just not enough. So what we in others are proposing is to go towards a multiverse analysis, where the same data are being analyzed with different pipelines, reasonable, justifiable, validated pipelines, but different. And then in kind of a meta-analysis, we could test whether the results converge, and then we could know if a specific result or conclusion is robust to different analytical choices or not, and maybe we could learn more from that. Tools are starting to being developed for this, but it's it takes time. It's really hard computationally and methodologically. So it will take some time, but there are starting to be some tools. You could read about them in the paper in our review preprint. But hopefully in a few months or years, we will have better solutions that are scalable and robust, and people could use them to actually do it. And we also need to figure out how to report it in a manuscript, and just adapt. So hopefully this is the future, but it will take some time. And I will go back to Guiomar.
So finally, we reached the research dissemination section, when we will show how to share your data, your code, your derivatives, and then your paper publication. And most important is that the research cycle doesn't finish with the paper publication. You can go beyond that and disseminating your results and outreach. So let's start through some basic, through the research cycle. We produce multiple outputs that far beyond publications, and each of them can have different levels of reproducibility and openness. So for them to be useful when they are shared, they need to follow the FAIR principles, which are data should be findable, accessible, interoperable, and reusable. Findable using persistent identifiers like DOIs that point you directly to the data you want to share. Also to have rich metadata and to describe the dataset, and so once you find it, you are able to know what's in there, and that they are indexes in a searchable resource. Then we need the data to be accessible, which is shared in a public resource with open or controlled access, depending on the regulations, and interoperable, that is following a common standard for organizing your data or your vocabulary, like BIDS, so anyone can take it and use it in their pipeline easily. And finally, reusable, which is linked to all of these together, which is really described, so people can understand what's in there, with detailed provenance of what's the history of this data, and an appropriate license that indicates what are the permissions that you concede to the user to be reused or openly or modified, and this. Sometimes licenses are forgotten, but this is a very important part on data sharing. For data sharing, your raw data, as we have seen, data have to be shared following the FAIR principles, and it has multiple benefits, and sometimes it's now required by some journals and funding agencies. Open data, when we refer data sharing, doesn't mean to be completely open share data with uncontrolled access, because sometimes the regulation doesn't allow you to have completely open data. However, it can be public and in a public repository with control access. This is also important for data sharing. There are many free Neuro-Imaging data sharing platforms, each with some degrees of control over your data, like Open Neuro, which is completely open, and Brain Life, where you can have some control access, Gene, DANDI, which is the Distributed Archives for Neurophysiology Data Integration, the INDI, which is the International Neuro-Imaging Data Sharing Initiative, NIDRC for tools and resources. And there can also be some institutional and funder archives like the NDA, the National Institute of Mental Health Data Archive, or some platforms like the Canadian Open Neuroscience Platform Comp or eBrains. There are many free Neuro-Imaging data sharing platforms, and there are also many dedicated repositories like the UK Biobank, which hosts a specific data set from a project or an initiative, and they have their dedicated servers, like the Human Connectome Project, HCP, or the CAMCAM, or the Open MEG Archive, the EBCD. You will see there are many, and there are also some generic archives that are not Neuro-Imaging specific, like OSF, Zenodo, GitHub, or Fixair, where you can also share your data. Together with the data, sometimes it's good to publish data descriptors, which are not intended to communicate findings, research findings, but detail information about a data set along with the shared data. That means how you collect it, what includes, how to be used. And there are also some initiatives to recognize data sharing efforts, which is very good because this encourages people to do it more. And sometimes the open science practices adoption is limited by the incentives researchers receive, but there are some little like open science badges in journals or some prizes like the OHP data sharing prize, which at least gives you some recognition to your effort. Regarding the code or how we obtain methodological transparency, documenting analysis step is key for reproducibility, as we have seen. However, the journal method section sometimes is not enough when you have a very long process or your word limits. You have word limits in the journal, so the code is an accurate source of the exact analysis steps followed. So code should be organized and clear. Otherwise, the results might not be reproducible or even correct. And however, as we were saying before, sharing imperfect code is still much better than not sharing at all. So yeah, we really encourage sharing the code. And regarding this, there are some tools that are based on GIS, so user interface, which are very nice and interactive. You just have to click buttons, but this is a bit challenge to report the processing steps you have followed. So we have to pay a lot of attention, and it's very good that some of these tools now have the option to track data history and provenance, so you can look of what clicks you did to report accurately your results. Again, that code should be shared following the FAIR principles, and it's really important, as Rotem was saying before, when we want to share code, we have to ensure some version control either with Git or GitHub or any other tool, and have a permanent DOI, a permanent identifier like Zenodo or Brainlife or Software Heritage, where you just point to the exact code that you have run on your analysis, and yeah, not get missed with any posterior modification or modification. Derivative data sharing, which is how you share your results when they have been processed. And this is critical yet very challenging because today there are not many available tools or mature specifications for advanced derivatives like connectivity, for example. So the community lacks clear guidance and tools for that. There are some initiatives that are starting to get more traction, like BIDS. They have released a general specification for common derivatives. However, they are now currently active working for extending the advanced derivatives for the different Neuro-Imaging modalities. That is, you have some general guidance for general sharing. However, still some work needs to be done to have a really useful thing for people to share more specific things. And there's work on that, so we encourage you, if you have some ideas or some energy, to put some cycles there and help this to be fixed or finalized. And then there are some specific open Neuro-Imaging repositories, like for where you can share and derive data. This is mainly for MRI and some PET, like for example, NeuroVolt. Allows you to share statistical maps and parcellations and atlases from MRI and PET, or template flow, which allows you to share some templates and spatial maps across standard spaces, and BrainLife, which has some integration of data processing and apps and derivatives. And also, it's really important to think of other additional materials like tables, figures, slides, which also should be shared fairly. And with this, we go to the publication. The scientific papers are currently the most important means for disseminating research results. However, so it's very important to report them properly, and there have been some community efforts for guidelines and best practices, like the OHPM COVID for MRI or for MEG and EEG, and some independent effort for guidelines to develop guidelines for PET. And for in the EEG field is the Artemis, which is the agreed reporting template for EEG methodology, an international standard which allows you to, report your steps in your papers and which things you have done. Most of these have also associated web-based apps which facilitates the task for reporting. Papers should be accessible, preferable to everyone, so many scientific publications are hidden, unfortunately, are hidden behind paywalls. And this is slowly changing, so both researchers and funding agencies are pushing towards open access. However, the adoption of these practices by major publishers may be considered arguable because of the pricing and tracking technologies and all this. So one solution or one option that also we can have is to publish preprints, which are publications prior to peer review in scientific journals. So they become publicly available, and this accelerates the release of new findings, allow early feedback prior to final publications, and allow an open access to everyone. And there are other initiatives like Open Research Europe, which allows you to publish openly, or open community reviews for preprints, and this. As well as this, there are now also starting to appear some hybrid publication formats in which you can include text, code, data, and runtime environment. So you can not only have your texts with the explanation of your results and your scientific hypothesis, but also you can run and reproduce the exact results you show. And some, like Neurolible, is a very nice initiative of this kind. And some traditional publishers have partnered with companies like Code or Cyan to have this. And a reminder that the research cycle continues beyond paper publication, so dissemination and outreach is really important in workshops, conferences, social media, newspapers. And finally, we have experienced a really shift in the way of doing research. So now, rather than carrying out a study from inception to results and only then disseminates findings, researchers now get multiple opportunities to share their ideas and results as they are being developed. Like from the pre-registration to each of the steps, as we have seen throughout this talk, we have opportunities to openly share and get feedback. So this provides a more transparent and inclusive and collaborative process. And we should aim also for equity, diversity, and inclusivity, creating welcoming spaces from everyone. So finally, as we have seen, there are really abundance of open tools and practices, which is promising and challenging at the same time. We try to give our expert-driven integrated review of the things that we knew. And we really encourage you to check out the preprint for much more information. And we have in the preprint also an additional resource table with an additional material where you can find links to all these resources and useful tools that we mentioned, so you can easily access to them. So I really want to thank you, and yeah, if you have any question now.
Thank you very much for this wonderful talk. It must be super useful for neuroimaging researchers who are interested in open science but don't know how to start, and also for people like me who is already working on open science but haven't got the comprehensive information at different stages of research cycle. And it is also very good to know that we have so many steps that we can take, and we can take the step one by one. Thank you. Maybe I can be a bit selfish to ask a question for myself, because I already have a lot of questions in my mind. So I have a question about the data pre-processing, because that is also the questions that I'm having now. So I think data pre-processing is a big issue in terms of the reproducibility, because there's such a big difference between different groups and in how to pre-process the data, and of course different atoms across studies. I think the ideal situation will be that we all use the same standardized pipeline. That would be but I think that would be very difficult to achieve. So I was wondering, with the reality that there's such a big difference in pre-processing and of course other steps, and we cannot change it in the very near future. What else we can do to make our studies more reproducible across different pre-processing pipelines? I think it's a very good suggestion from your presentation that about the multiverse analysis, but I was also wondering about the feasibility for the junior researchers like me to do it for every project. So my question is basically, is there a way for junior researchers to do transparent research with so many restrictions, and knowing that the gaps between the reality and the best way that you presented?
I think I'll try to answer that. Yeah, okay, this is a great question. So I think there's a lot to say about that, and I would separate between two things, like reproducible in a way that like can you reproduce the result from the same data, the same pipelines, can you get exactly what you got? So this is really not much dependent on how many choices or available software there are, as long as you report exactly what you did and give your code, and or if you have one, or just say exactly transparently what you did, your result should be reproducible, or your pre-processing should be reproducible from the pipeline. I completely agree with the issue you raised, right? Because I told you about a paper that we did about that, and there's a problem that different choices may have led to different results, and pre-processing is a huge part of that, obviously. I agree that there are some efforts to standardize. It would help, but I don't think there's an ideal pipeline that everyone should use, because it depends on your specific research question code, and there's not one golden pipeline. So this is why I think multiverse is the answer. I agree that now for junior or senior, by the way, researchers, it's not feasible yet. I'm a postdoc too, so I'm an ECR too. I know it's not feasible yet, but I'm really hoping that the many efforts that are put into this would yield tools that each of us could use, on our level, to actually do it in each single study. I think for now what you can do is make sure that you're transparent, and you could reproduce your results and you're clear about it. And maybe if there's one specific choice and you're not sure about, maybe you could try two different things and report both of them. These are important, no picking, just report everything. And this could be useful. I saw it like when I review papers, even I see it sometimes that people are starting to do that. So I think we're going to this direction, at least in MRI.
Yeah, thank you very much for your answer. That's also what I'm trying to do to make my code and also my results, are marked down, this kind of thing, to make my research as transparent as possible. And I hope that in the future we can move towards together to the ideal. Yeah, and I see that we're just on time. And do we have more other questions? No, I didn't see other questions from the audience. So then thank you very much for the great presentation discussion. It is really an enjoyable 1 hour with tons of information. Hope this talk can encourage more neuroimaging researchers to start open science practices. Thanks again for being here, and hope to see you next time. Have a great evening.
Thank you for having us.
And maybe
thank you.